Good morning, everyone. Thank you for being here today. I'm excited to talk to you about an essential aspect of cell biology, endocytosis and exocytosis in cells, and how these processes help cells interact with their environment. But before we dive into the specifics of these processes, let's first do a quick review of basic cell structure and the cell membrane, as this will help us better understand how endocytosis and exocytosis work. The cell membrane is the protective boundary of the cell. Think of it as a border guard that controls what enters and exits at the cell. It is composed of a phospholipid bilayer, two layers of lipid molecules that form a semi-permeable membrane. The bilayer has hydrophobic or water-repellent interior layers and hydrophilic or water-attracting outer layers. This structure allows selective permeability, which is crucial for maintaining the internal environment of the cell. Embedded in this membrane are proteins that act as gates, pumps, and receptors for different molecules. Channel proteins form passageways for molecules to move in and out. Carrier proteins help in active transport, moving molecules against their concentration gradient. And receptor proteins bind to signaling molecules like hormones or neurotransmitters, allowing the cell to respond to internal or external signals. We will also review on the transport across the cell membrane. Transport can be passive or active depending on whether energy is required. With regard to simple diffusion, small molecules like oxygen or carbon dioxide pass freely through the membrane, moving from areas of high concentration to low concentration. Now, what about larger molecules or charged particles? Larger molecules like glucose or charged particles like ions cannot pass directly through the lipid bilayer due to their size or charge. These molecules require more specialized transport mechanisms like endocytosis or exocytosis to enter or leave the cell. We'll first discuss endocytosis, which is the process of bringing substances in. Endocytosis is the process by which cells engulf large molecules or particles by forming vesicles from the plasma membrane. It allows the cell to take in materials that are too large to pass through the membrane by diffusion. Types of endocytosis include phagocytosis or cell eating, pinocytosis, which is also called cell drinking, and receptor-mediated endocytosis. Phagocytosis is the process by which a cell engulfs large particles such as debris, foreign substances, or microorganisms. The cell extends its membrane around the particle, forming a phagosome or a vesicle containing the ingested material. This vesicle then fuses with a lysosome for digestion. Examples are white blood cells like macrophages use phagocytosis to remove bacteria and pathogens. Pinocytosis is also called cell drinking. Pinocytosis is the process by which cells take in extracellular fluid and dissolve solutes. The cell membrane forms small vesicles around the fluid, bringing the substances into the cell. Example is, this process is important for nutrient absorption as it allows cells to take in essential nutrients from their surroundings. And last, under endocytosis is receptor-mediated endocytosis. Receptor-mediated endocytosis is a more specific form of endocytosis, where the cell surface contains receptors that recognize and bind to specific molecules, what we call ligands. Once the ligand binds to its receptor, the membrane forms a vesicle that contains the ligand and its receptor. Example is, this process is crucial for the uptake of cholesterol in the form of low-density lipoprotein or LDL. Now let's move on to the key steps in endocytosis. Step 1. Recognition. Receptor proteins on the cell membrane recognize specific molecules. Step 2. Membrane invagination. The membrane folds inward, forming a vesicle around the substance. Step 3. Vesicle formation. The vesicle pinches off from the membrane, carrying the substance into the cell. And step 4 is fusion with lysosome if needed. The vesicle fuses with a lysosome for digestion or processing. Now we're going to talk about exocytosis, or the process of releasing substances. Exocytosis is the process by which cells expel materials by merging vesicles with the cell membrane. This mechanism is essential for removing waste products and releasing substances like hormones and neurotransmitters. Its role in the cell include exocytosis helps maintain the balance of molecules inside and outside the cell and allows for communication with the surrounding environment. And now we're going to talk about the key steps in exocytosis. Step 1. The vesicle formation. Materials to be expelled are packaged in vesicles. Step 2 is vesicle transport. The vesicles move toward the cell membrane. Step 3 is vesicle membrane fusion. The vesicle fuses with the plasma membrane. And step 4, the release. The contents of the vesicle are released outside the cell. And now we're going to talk about the examples of exocytosis. Number 1 is neurotransmitter release. Neurons use exocytosis to release neurotransmitters into synaptic clefts, allowing communication between nerve cells. The next one is secretion of hormones. Hormones like insulin are released from endocrine cells through exocytosis into the bloodstream, where they can affect distant target organs. And last example is waste removal. Exocytosis is used by cells to expel waste products, ensuring the cell maintains homeostasis. We're going to talk about the importance of endocytosis and exocytosis. First is in cell communication. Both processes are essential for cell signaling and communication, allowing cells to interact with their environment, receive signals, and respond accordingly. Next is nutrient and waste management. Endocytosis allows cells to take in nutrients, while exocytosis helps remove waste, maintaining balance and efficiency. The third one is immune response. In endocytosis, cells in the immune system such as macrophages and dendritic cells use phagocytosis to engulf and digest pathogens like bacteria, viruses, and dead cells. This is crucial first line of defense. In exocytosis, after processing pathogens, immune cells use exocytosis to release cytokines, signaling molecules that help coordinate the immune response by recruiting other immune cells to the site of infection. And lastly, homeostasis. By controlling the intake and release of molecules, these processes play a vital role in maintaining a stable internal environment, supporting cellular function and survival. We also talk about its application. First is in diseases linked to endocytosis or exocytosis. First is virus and endocytosis. Many viruses rely on endocytosis to enter host cells. Viruses such as influenza virus, HIV, and coronavirus bind to specific receptors on the cell membrane. Once bound, the virus is internalized by endocytosis and its genetic material is released into the host cell to hijack the cell's machinery for replication. With regard to cystic fibrosis and exocytosis, cystic fibrosis is a genetic disorder that affects the movement of chloride ions across cell membranes. This disrupts exocytosis and affects the secretion of mucus, causing it to become thick and sticky. The abnormal exocytosis of chloride ions lead to the buildup of thick mucus in the lungs and digestive system, making it difficult for the body to clear infections and leading to respiratory and digestive issues. With regards to research applications of endocytosis and exocytosis, first is drug delivery systems. Endocytosis plays a major role in drug delivery systems, especially in targeting specific cells or tissues. Nanoparticles, liposomes, and other carriers are designed to exploit endocytosis to deliver drugs directly into cells. By mimicking the cell's natural processes, researchers can create more efficient and targeted therapies. With regards to vaccines and exocytosis, exocytosis is a crucial process in the immune response triggered by vaccines. Vaccines often work by introducing antigens in the form of weakened or inactivated pathogens into the body. These antigens are taken up by immune cells via endocytosis. Once processed, they are presented on the surface of immune cells and trigger an adapted immune response. In summary, endocytosis and exocytosis are essential cellular processes for transporting large molecules, maintaining homeostasis, and facilitating communication with the cell. These processes are crucial for various functions such as nutrient uptake, waste removal, and intercellular signaling, highlighting their importance in cellular health and function. Thank you for your attention. See you in the next module. Bye-bye.